All righty, are we good?
All set.
Okie dokie. So I'm going to give a kind of a broad overview of some of the ongoing research that is taking place in my lab. The title of my talk, Trait Variation and Covariation in Long-Lived Plants, Understanding Form, Function, and Phenotypic Plasticity to Advance Food and Ecosystem Security. So I'd like to start just by thinking broadly about what sustainable ecosystems look like in nature. And whether we're in the jungles of Costa Rica, as in here in Manuel Antonio, or in the deserts of the Southwest, all these different ecosystems consist primarily of long-lived plants living together in mixtures, not exclusively of long-lived plants, but primarily. Even here in the Midwest, this is the Kanza Prairie in central Kansas. So while our natural ecosystems are comprised primarily of these longer-lived plants living in mixtures, when we look to agriculture, the vast majority of contemporary agriculture, again, not entirely, but most of it is made up of shorter-lived annual plants grown in monoculture. And so there is a major difference between the attributes of what have evolved to be sustainable ecosystems in nature and what we are attempting to make sustainable in agriculture. So the first question that motivates the work of my lab group is, can perennial plants have a prominent role in agriculture? And there are many perennial plants already that were domesticated thousands of years ago. One example I'll talk about today are grapevines. But the majority, and in fact, I would argue all of the perennial plants that were domesticated historically were perennial woody plants. But there are thousands of perennial herbaceous plants out there that have not yet made it or are just beginning to enter the domestication pipeline. So if we focus for a moment on perennial woody plants, we can say that the domestication process in these woody plants has been unique relative to what we understand about how domestication happens in annual plants. So this figure from Brandon Gott's 2016 Trends in Genetics paper identifies on the left the wild ancestors of perennial crops and notes that most of these populations are primarily outcrossing. Many are even dioecious, that the primary mode of reproduction in many woody perennial crops is clonal propagation. It's estimated about 75% of woody perennial crops are clonally propagated, that there are cycles of introgression and hybridization, often with wild relatives, with other species, and then additional clonal propagation that ultimately leads to a population of cultivars, clonally propagated cultivars, that are often highly heterozygous, but carrying some significant genetic load. This is our current understanding of how domestication has happened in perennial woody plants. Now, interestingly, we know very little about the process of domestication in perennial herbaceous plants because it has really only started relatively recently. So what my lab is interested in doing is asking basic questions in plant biology, perennial plant biology, and evolution, just to advance our understanding of how these plants live and respond to their environments, how they alter their morphologies to deal with seasonal and annual variation and all sorts of biotic and abiotic pressures, the genetic underpinnings of some of those traits, and then how that information can help guide and optimize domestication processes, both in woody perennials and in what are just entering the pipeline now, perennial herbaceous species. Okay, so to think about trait variation and covariation in long-lived plants, I do want to emphasize this woody perennial dichotomy. So the first thing I'm going to talk about today is some of our ongoing work with grapevines, and then I'm going to move into our work in perennial herbaceous species. So The first question that we're asking, and kind of a major area of work in the lab right now, is how root systems shape shoot system variation in long-lived plants. We're going to talk about grapevines as a model, grafted grapevines as a model for understanding this question. And then we'll shift gears a bit and move into some work in perennial herbaceous species to ask how traits vary and co-vary over long lifespans, and how whether or not traits that we see early on in the lifespan of perennial plants can actually predict variation in mature plants. And then the last thing I want to talk about is how perennial plant traits change in the context of plant communities and the soil ecosphere, and how do those traits, what role they play in actually altering the communities that they live in and the soil ecosphere. And we ask all of these questions with an eye towards application, primarily in agriculture. So I'm not a plant breeder, but I work closely with breeders on all of the projects that I'll talk about today. And really, again, we're just trying to understand basic biology that might help underpin, might help inform some of the breeding processes. Okay, so first, how do root systems shape shoot system variation in long-lived plants? This is a plant genome project we've been working on for 6 years now, and we're using grafted grapevines to try to understand that relationship between root or rootstock and shoot or scion. So in grapevines, most commonly, the above ground part of the plant, the scion, is derived from a European species, the only European grapevine species, Vitis vinifera. In eastern and midwestern parts of North America, we're growing primarily hybrid vinifera, hybrid Vitis hybrid species that are often have quite complicated pedigrees with 5 or 6 different grapevine species in them. Side note, there are 19 native grapevines in North America, and they are all, with one exception, interfertile with the European species. Now, the below ground portion of grapevines, the rootstock, is usually an F1 hybrid that is the result of a cross between 2 of the 3 species I'm showing here. Vitis cinerea variety helleri, this is a native species from Texas. Vitis riparia, which I think you probably have in your area, the riverbank grape, and Vitis rupestris, the rock grape, which the final 2 of those occur here in Missouri as well. Okay, so to understand this question of how what's going on below ground in perennial plants affects above ground phenotypes, we've been working in an experimental vineyard at the University of Missouri's Southwest Research Station in Mount Vernon. And in this experimental vineyard, we have a local cultivar, a red grape cultivar named Chamberson. This is a hybrid grapevine, which is growing ungrafted, so growing on its own roots. And then we have it grafted to 3 different rootstocks. The grapevine rootstocks have these funny names like SO4, 3309C, and 1103P. They're all grafted with Chamberson, so the leaves of the vineyard, the above ground parts of the vineyard are all the same. The below ground components are one of these 4 options. So our team went into this vineyard 3 years in a row, 2017 through 2019, and we sampled leaves, Chamberson leaves, at anthesis in mid-May, at veraison when the berries are shifting from green to red, usually in late July, and then at harvest in mid to late September. And we collected samples and phenotyped everything we possibly could. And I'll give just a quick overview of what that is, but I'm only going to talk about one trait today. So we looked at for Chamberson, we looked at the shapes of the leaves. We looked at the elemental profiles of the leaves, what ions are in those leaves. We looked at gene expression patterns in both the leaves as well as the reproductive tissues. We measured vine physiology. We measured berry chemistry, and then also in some cases, wine volatiles. We also looked at the vine microbiome as well as the Chamberson epigenome. So I just wanted to pull out one example here, the ion concentrations in the leaves. So this is the work of Zach Harris. Zach is a PhD candidate here at St. Louis University, getting set to graduate this May. This paper, Root System Influence on High Dimensional Leaf Phenotypes over the Grapevine Growing Season, includes a section on what we call the ionome, or the elemental profile of the leaf. Now, the reason we're so interested in these ions is that it's through the root system that ions enter the plant. And we quantified 20 different ions in the leaves of Chamberson, and then asked how those ion concentrations varied as a function of the rootstock to which that Chamberson vine was grafted. So the first figure I want to show is a heat map that shows along the kind of the side left side here, the 20 different ions that go from aluminum down to zinc. And then going across the top here is the factor that we were considering and asking how much of the variation in the concentration of any particular ion was explained by each of these factors. So we looked at the leaf position, which is the position of the leaf along the vine, whether it's a very old leaf or a middle-aged leaf or a young leaf. We looked at the phenology, that's the time in the year when the leaves were sampled in the spring or middle of the year or in the fall. We looked at the rootstock as a potential factor affecting concentrations of ions, and then we looked at all of those interactions. There was also an irrigation treatment within this vineyard. However, in the year, these data are just from the year 2017. It was a really wet year, and so in Missouri, there was not much of an effective irrigation. So what I wanted to point out is first, the position of the leaf along the vine, as well as the timing of the sampling, is those are the factors that explain the most variation for most ions in the leaves. But the rootstock, as you'll see shown kind of here in yellow and in some cases orange, explains a significant portion of the variance for all of the ions in this rootstock column for which there is a color other than white. And I'll point out that magnesium, molybdenum, and nickel all have a relatively high percentage of variation explained by the rootstock. Another interesting thing about these data is that we can look at the concentrations of specific ions and how each of the rootstock, what effect it has on the concentration of that ion in the leaves. So again, looking at anthesis, veraison, and harvest across the span of the season, it's interesting that Chamberson leaves grafted to 1103P have higher concentrations of cobalt in the spring. In the middle of the year, SO4 also confers that higher concentration of cobalt, but by the end of the season, we're seeing that kind of reduced as uptake through the roots is decreasing with pending senescence. With nickel, we see a different effect. So it's SO4, that is taking the rootstock SO4, which is leading to higher concentrations of nickel in Chamberson leaves grafted to SO4. And that's an effect that we see consistent through the first 2/3 of the growing season, and then it falls off at the end. The last thing I want to show in these data are that we can actually use these machine learning confusion matrices to ask whether or not we can predict, based on the ion concentrations of the leaves, the rootstock to which that vine was grafted. And this kind of blue diagonal shows here that we can actually accurately predict rootstock based on ion concentrations, and that the accuracy of that prediction increases when we take into account the season, the time of the season in which the samples were collected. The last figure on the right looks at our ability to predict the position of the leaf on the vine based on its ion concentrations, and we can do that as well. So this trait is really powerful in terms of essentially a proxy for what is happening below ground. And we have another paper we're just wrapping up now that looks across the 3 years of our sampling, and the temporal, both seasonal and temporal aspects of ion uptake by rootstocks it's just such a dynamic and interesting process. So to try to understand a little bit about the genetic basis in the root of some of the traits that we're seeing in the shoot, remember the ion concentrations I just showed are one of many traits that we're looking at. We've developed a replicated rootstock mapping population. So we crossed 2 of the species that are commonly used for rootstocks, made an F1 population, and then grafted to that F1 population a common scion. We just planted this out in St. Louis last summer. So this is the rootstock experimental mapping population that we'll use essentially in a QTL study for many of the traits that we're looking at. But we know that the environment plays a huge role in this. So we've replicated that planting. It's in St. Louis, but it's also in Springfield, Missouri, in Brookings, South Dakota, in Geneva, New York, and in State College, Pennsylvania. And the kind of the long-term goal of this work is to see if we can optimize grapevine growing and adapt grapevines for future climates from the roots up. And so in a vineyard like this in southeastern Missouri and St. Genevieve, can rootstocks be used to optimize this particular vines for this particular region? And we think this is a relatively underexplored area, particularly in the Midwest, for future research. Okay, so I'd like to switch gears now and move away from our perennial woody plants. Grafting is a very common phenomenon in perennial woody crops. It's like about 70 major perennial woody crops are grafted, but that's really not the case in most herbaceous perennial plants. And so the questions we're focusing on in these perennial herbaceous species have really more to do about relationships among traits. How do traits vary and co-vary over the course of a lifetime of a plant? And can we predict from early life stages traits that we'll see later on in life? This is a classic strategy, early stage selection, but we're applying some high dimensional phenomic data to try to build on previous work. So why is trait variation and covariation important in perennial herbaceous species? And why is it important as we think about perennial herbaceous species and their future role in agriculture? I have a long-standing relationship with the Land Institute in Salina, Kansas, which some of you might be familiar with. In fact, I know some of you are. This is a tremendous organization that is working to transform agriculture perennially, as they say. And they're working to develop perennial herbaceous species that can be grown in polyculture to essentially reverse the degenerative trends of some agricultural practices today, and through their large root systems, help reverse those soil trends and in fact build soil through perennial polyculture agriculture. So there, as I mentioned before, there are no perennial herbaceous crops that are grown for food that I am aware of that have made it into large scale commercialization. And the Land Institute's breeders are working to change that. So there's one species that is particularly far along, relatively speaking, in the breeding process, Thinopyrum intermedium, or intermediate wheatgrass. This is goes by the trade name Kernza. The Land Institute, this is their image. It's a famous one. This juxtaposes this intermediate wheatgrass next to an annual wheat plant. And you can just see the differences in these root systems. It's quite dramatic. So this is one of several species that the Land Institute is exploring. And our role in this work is again, to try to understand some of these basic biological questions that might help guide some of the breeding. Now, if you're curious about Kernza, if you've never heard of it, I wanted to share that you can buy Kernza flour and grain on Amazon. You can buy it from Perennial Pantry. We use it in pancakes. It's really delicious. So if you are perennial curious in terms of grains, I I recommend giving this a shot. Interestingly, Patagonia Provisions just recently came out with a Kernza pasta. I just got it this week, but I haven't yet had a chance to try it. Alrighty, so back to the science. One of the first things we wanted to ask is, okay, if herbaceous annual plants were domesticated and have been domesticated for thousands of years, but perennial herbaceous plants haven't, is there some like inherent difference in these two types of plants? And perhaps that's one of the reasons why early farmers didn't domesticate perennial herbaceous species. So the first question we wanted to ask is, how do traits vary and co-vary in annual and perennial congeners? So we picked 3 genera, legume genera, Lathyrus, Phaseolus, and Vicia, and we compared early life stage traits, things like seed size and shape, germination probability, and growth rate, to see if they differed among the annual and perennial members of these 3 genera. This work was carried out by Sterling Heron. Sterling completed his PhD last August at St. Louis University. He's now working as an ecologist in Florida. But he published a paper in the American Journal of Botany where he looked at, again, aspects of the seeds, of germination probability, and of growth rate, and was able to demonstrate that genus is the strongest predictor of both seed size and shape. So in this middle panel, the different genera are coded as different colors, and they are in fact statistically different from one another. But its lifespan, whether the individual species were annual or perennial, that consistently predicted both plant height and leaf number. So there are some differences among the annual and perennial members of these genera. This study only looked at the earliest life stages of these plants, but it did give us a chance to also explore trait covariation. So this very colorful figure looks at seed traits on the left. So this pentagon here is several different seed traits, which are very positively correlated to one another, as shown by these strong blue bars. But interestingly, seed mass, perimeter, length, area, and width are negatively correlated with the shape of the seed roundness and circularity. That's when we look across the full data set. What was interesting in this study, though, is when we compared the annual members of these 3 genera with the perennial members of the genera, we see that seed traits generally co-vary with aspects of growth and height, which are shown on the right in annual species. But the perennial species, we see kind of a decoupling of the seed traits from the germination and height traits. So this might be important from a breeding perspective, because these data suggest in the earliest stages of the life here that there is a relationship between traits in annual species, but not in perennials. We looked across the different genera as well, and although they are unique, we didn't see any major standouts here. All right, so we wanted to expand this study by then asking, what is the relationship between traits that are expressed early in the lifespan and those appear at much later stages? So Sterling's study basically stopped at around 8 weeks of growth. But for this, we wanted to look across multiple years. So we put together a panel of about 12 different perennial herbaceous species, some of which are known candidates for perennial crop development, like Thinopyrum intermedium, Silphium integrifolium, Anabricus vicifolia, and others of which are on our radar, but a little earlier on. And what we did is that this next slide's a little bit big, we started carrying out high throughput phenotyping in the earliest stages of the life cycle here at the Danforth Center. So a little bit like what Sterling was doing, we measured seed weight and morphometrics, germination timing. We looked at plant size and growth rate using an automated phenotyper I'll show in my next slide. We also looked at a number of spectral traits We've looked at the ion concentrations of the leaves, as well as HSV color and NIR profiles. So one of the cool aspects of this project is that we put these 12 different species on this phenotyper at the Danforth Center, where we measured everyday growth through top-down photographs and side view photographs, both with RGB and with NIR imaging. I'm super proud to say that we more than doubled the number of species that had ever been on this facility in our first run. Again, this allows us to measure growth rate over time. All right, so for one aspect of this study, we looked at accession level variation across 5 weeks. So we were estimating phenotypic traits related to plant size, shape, and color, and use those to estimate growth. So shown here are basically two individual Silphium plants. There were 2,280 plants that went through this on the phenotyper. And you can just see that there's quite a difference in size, and you can eyeball it, a difference in growth rate between these two accessions. We were also able to demonstrate differences in hue, saturation, and the value. These are different metrics of spectral signatures. And what's really cool is we're beginning to see accession level signatures in these different spectra that we are measuring. So why does any of this matter, and what are we doing with these data? So we have these early stage traits, seeds, germinations. We did look at root and shoot biomass for a subset of destructively harvested samples, the side view and top down images and the spectral signatures. We measured all of these traits, and then we threw our plants into a U-Haul, and we took them out to our field site where we planted them in a spaced plant nursery. And there we began collecting data on flowering time, I should say on overwintering, and then flowering time, height, and then ultimately several different yield components. And what we're trying to see, again, if there's a relationship between these early stage traits and traits that we see in the field, and if that relationship is consistent from one species to the next. So I'm going to show a series of figures now that are correlation matrices that show the relationships between different traits. This is just an example with some seed morphometric traits from Silphium. So whenever you see a blue square, then the traits are significantly positively correlated. And the darker the blue, the stronger the correlation. When you see a red square, the traits are negatively correlated. And again, the darker the shade of red, the more strong that negative correlation is. And for the white squares, there's no significant correlation. So first, if we look across all of the species for which we've collected data, we are seeing strong patterns of both positive and negative correlations across species. So this figure shows the early life stage traits that we've measured here at the Danforth Center on the left, going from seed weight through germination, leaf number, and then some spectral signatures. And then looked at the relationship of those to some later life stage traits, including overwinter survival, flowering time, height, yield, and the proportion of viable seeds. So this indicates that there is something to these early versus late life stage relationships. So just as an example, I'm going to focus on Silphium integrifolium and demonstrate that early life stage traits measured under controlled conditions at the Danforth Center correlate with traits in the field. So a couple of examples. If we look here at seed weight, area, length, and width, we can see that traits of the seed are negatively correlated with incidence of orange rust later in the field that we saw at that were planted out at our field site. They're also negatively correlated with the number of heads that form. They're positively correlated with the number of seeds and the number of what we call good seeds, or seeds that filled. We can look then also at the area. So this is a measure of the size of the plant and growth rate, and see that the size of the plant in its seedling stage positive is negatively correlated with the days it takes to flower, but positively correlated with the height of the plant in the field. We also see a relationship between growth rate and several metrics of yield. Okay, and then if we lastly look at some of our spectral signatures, here looking at hue, value, and saturation, where we see a negative relationship between hue with height of the plant in the field and also head number, as well as a relationship between saturation and then the total number of seeds produced by that plant. So this is inherently biologically interesting for people who like to think about long-lived plants, as I do. But I think there's a real practical application to this information. So we have a new project going from the Foundation for Food and Agriculture Research called Reimagining Crop Domestication in the Era of High Throughput Phenomics. So for this, we're focusing on 3 of those species, intermediate wheatgrass or Kernza, sainfoin, which is Anabricus vicifolia, and then Silphium. And we're asking, can elite individuals of perennial species be predicted by phenomic relatedness models based on seedling and other high dimensional traits that I've just described? And we're also comparing for Kernza phenomic selection with genomic selection. So we're interested if it is as effective or maybe even more effective to apply phenomic selection and relative to genomic selection. We're also doing the concomitant cost comparisons for Kernza. So where this project is now is that we had another round of plants. I'm trying to think, we're just in our first year last fall. We planted 2,000 individuals of sainfoin and intermediate wheatgrass. We measured these plants for spectral signatures. We put them on the phenotyper. We collected all of those early life stage traits. And then we planted the plants out in 2 locations, here in Salina, Kansas, and then also in St. Louis. So we have another round going out this spring and a couple more going out next fall, where we'll be able to test these hypotheses and hopefully evaluate the potential of these phenotypic traits to expedite the breeding process of these new perennial herbaceous species. Okay, I have one more thing I wanted to tell you about, just kind of briefly, which is we've talked a lot about now perennial plants. We've talked a bit about how roots relate to shoots, and then we've looked at patterns of trait covariation across the lifespan of these plants. But where we're going next is to put those perennial plants in the context of their communities and the soil. And so this is a new project called the New Roots for Restoration Biology Integration Institute. It's funded by National Science Foundation. We have about 40 individuals and 9 institutions participating in this project, including the University of Vermont and Eric von Wetberg. So as we saw in the beginning of this presentation, most natural ecosystems consist of long-lived plants that are growing together in mixtures. And perenniality and diversity really are hallmarks of all of these systems. So when we think about how we might go about restoring or restructuring ecosystems, and it's estimated about 75% of land consists of degraded ecosystems. We're thinking about how we would do that based on the interaction of perennial plant traits with their environment. So the Institute is focused in part on how plant traits relate to the environment with an eye towards restoration of, we'll call them natural ecosystems. But we're also thinking about how we can use our understanding of plant traits and the environments to reimagine agriculture, and specifically to think about perennial polyculture agriculture that might mimic these perennial and diverse natural ecosystem. So here we have Kernza intermediate wheatgrass growing in an intercrop with this perennial legume. It's just with alfalfa. So the whole goal of the New Roots for Restoration Biology Integration Institute is to put our understanding of the plant organism, both above and below ground components of that system, in the context of plant populations and plant communities, and to place that in the context of the soil ecosphere, both the abiotic and biotic components of the soil. And we're doing this to ask two big questions. One, how does the soil ecosphere and the plant community influence above and below ground variation in plant traits? But perhaps even more important than that is the following question: how do above and below ground plant traits influence properties of plant communities and the soil ecosphere? And if we can understand that, can we guide our selection of both plants to be used in restored areas, as well as plants to be included in perennial polyculture agriculture to guide the restoration of both our natural and agricultural lands? So the Institute has these three major themes: plant organismal systems, population and community ecology, and the soil ecosphere. And we've developed a series of 10 projects which are shown here by number, that were intentionally designed to integrate across these subdisciplines of biology. Well, one of the parts of this institute that I'm most excited about is that we are very intentionally applying the high throughput phenotyping that a lot of which happens here at the Danforth Plant Science Center. Technologies like above ground plant phenotyping, like root imaging via mesocosms or X-ray CT scanning, or this high throughput elemental profiling. These are approaches that have been developed primarily for and applied mostly in model systems and annual crops. And we're using these in 28 different perennial herbaceous species, some of which are potentially important for ecological restoration and or for perennial polyculture development. So one of the projects that has an outpost in Vermont I wanted to mention is this project 2.3. Do root shoot plant traits vary in monocultures versus polycultures of perennial herbaceous species? And does this change with the addition of a tree species? Related to that, how do monoculture and polycultures of perennial crops impact soil abiotic properties and soil community structure? How do soil communities differentiated by crop identity and diversity influence crop root shoot traits? So this is a project that would allow us to look at plant soil feedbacks on plant traits themselves. So to do this, we have in our institute, in our Biology Integration Institute, several partners in St. Louis, and kind of along this I-70 corridor and I-55 corridor, as well as the University of Vermont. We have planted out an experiment which has, I'm sorry, my slides are a little out of order here, which has a monoculture of intermediate wheatgrass, a polyculture of intermediate wheatgrass and alfalfa, as well as alfalfa by itself and intermediate wheatgrass with a fertilization treatment that was planted this fall in 6 different sites of the Institute. All right, so these are the different institutions that are participating within this. And what we're hoping is that over the coming 5 years, we'll be able to use that experiment to understand this relationship between plant root and shoot traits, how it varies in the context of a simple agricultural community experiment, and how that varies across sites. So please stay tuned. We're in month 4, I believe now, of this Biology Integration Institute. I wanted to share the mission of the Institute and also put in a plug for anyone who might be interested in this. So our mission is to advance scientific discovery to support the development of resilient, long-lived systems in nature and agriculture, In carrying out this work, we're training the next generation of diverse scientists to address global challenges of both food security and ecosystem function. We are recruiting now at all levels across multiple institutions, including an REU program and a high school program. So if you're interested, please visit our website or email me directly, and I'd be happy to share more about it. All right, so I'm going to go ahead and wrap up, and thanks very much for listening to this presentation. Today, we talked a bit about long-lived plants and focused on trait variation and covariation in a number of different systems. So we talked about using grafted grapevines to understand how roots affect shoots in long-lived plants. We looked to perennial herbaceous species to understand patterns of covariation across the lifespan of a plant, and saw some evidence that early life stage traits might be used to predict traits we see later in the lifespan of a plant. And then lastly, I introduced this Biology Integration Institute, New Roots for Restoration, which places perennial plant phenotypic data in the context of plant communities and the soil ecosphere, and which is really looking towards using that information to guide restoration in nature and in agricultural settings. I'd like to just end quickly by acknowledging I have a huge team of collaborators on each of these projects. Our grapevine team, we call ourselves Grapevitis Underground. You can follow us on Twitter or check out our website. Our perennial covariation team, which we call our PCOVAR group, is based here in St. Louis and also at the Land Institute in Salina. And then lastly, the Biology Integration Institute is growing by the day, but it's really a phenomenal and exciting team to work with. All right, I'm going to stop there, and I'd be more than happy to take any questions. Thanks so much.
Thank you so much, Allison. That was really insightful and very interesting. Does anyone have any questions? You can put them in the chat or unmute yourself. Eric. Oh, Jess.
Yeah, let Jess go.
Thank you so much, Dr. Miller. That was awesome. I have a lot of questions, but I'll just keep it to one. And one connection. Do you know about the SARE final report done recently in Vermont on sea kale, perennial crop, which takes Eric Tosminer's list, that book of perennial vegetables? If not, I'll put it in the chat. You can check it out.
I don't know it, and I would love to see it. So thank you.
Sure, great. My question is, I so appreciate your research and definitely feel like the importance of aspects of agriculture moving towards perennial systems with everything that's happening. And a lot of terms you talked about, like phenotype plasticity. I have a lot of curiosity about what that means. And I'm just curious, the most recent study at the end you mentioned, 2.3, I think, when you're looking at how soil structures changed, comparing perennials to monocultures to polycultures. Are you, some of the parameters you're looking at, are you looking at like fungal communities, like mycorrhizal communities? Just curious about that. Thank you.
Thank you for your question, Jess. And I'd be happy to talk about any of this anytime. And I know this was kind of a broad overview. So yes, we have a team within the Biology Integration Institute that is a microbial ecology team. And they are characterizing arbuscular mycorrhizal fungi, among others, in many, if not all, of the experiments, the 10 experiments that are part of the Institute. That group's based in Kansas. It's Jim Beaver and Maggie Wagner and Ben Sykes.
Great, thank you.
Eric, you're up.
All right. So I got looped into a fight that I thought had happened years ago. My master's advisor, Jake Weiner, got into a fight with Wes Jackson back in the 90s about whether or not an annual growth form was skewed in such a way that it would always outproduce perennials. And I thought that had gone away, but or I hadn't heard about that argument in a while. And it may just be the very large rock that I live under. But I saw, I want to say Bob Loomis had a paper that came out a couple of weeks ago that had basically the same complaint. And for that matter, an alum here who's at TLI, Chase Stratton, got this at the annual meeting when he talked about Kernza breeding, that a perennial growth form can never out yield or even approach the yield of a productive annual. Do you have thoughts on that?
Oh, I sure do. And a lot of people do too. And I think that there were a lot of fights in the 90s between Wes Jackson and, maybe ongoing. I think that what I am seeing is it's difficult to compare an annual plant with year one of a perennial plant. And most studies have done that. And I think that what we're learning is that we really have to look across years. Annual plants, their job is to get it done and get it done quick. And then they're done. And plants, perennials, they just don't do it that way. I mean, there's just a whole lot of other decisions that are getting made, other ways in which resources are being used, particularly in year one. So I don't buy that argument. I think it's kind of to be determined whether or not perennials in whatever year of their growth can outperform annuals. I would like to say one other thing, because this just went across Twitter yesterday, which is that Kernza, which again is this grain, this wheat relative that's the furthest along in the domestication pipeline, although I believe it's in cycle 9, so it's not that far along. Somebody was saying, well, it's still only producing at 30% of wheat. So what good is that? And the response of the Land Institute was, well, we're only in cycle 9, or what, we've only been doing this for a relatively short period of time. And so based on the current trajectory, given the yield gains that they've seen thus far in the breeding, they're estimating that they're going to hit the yield of wheat in a matter of, it's not years, but it's single digit decades. So I can't remember, maybe it was 20 to 30 years. So I guess the other argument against that perennials will never achieve what annuals can is they have not really yet been tried and are still very early on in that domestication process, even the ones that are furthest along. So I think it still remains to be seen. And I'm not seeing from our very early on studies an indication that this is not possible. I think it's probably not possible today, but I think it's possible. I haven't seen anything to suggest it's not possible ever. So.
There's a question in the chat from Jonna. Oh. Can I ask Jonna's question first? It was in the chat and then, yeah. So she asked, one of the ion graphs had a dark black box for phosphorus. What processes / interactions do you contribute to this finding?
Yeah, it's interesting. I don't have it right in front of me, but I think that was the dark black box was related to the position of the leaf along the vine. And so that means that, and I forget which one it is, but either the oldest or the youngest leaves have much higher concentrations of phosphorus than the other. And there are some ions that really have this very striking pattern, and it's different for different ions. Some are in higher concentrations in younger leaves, some are in higher concentrations in older leaves, but we've looked at this for hundreds of vines, and it's a consistent pattern for each ion. So I think that's what that is, but I can certainly direct you to the manuscript in which that is described, and also share a second one that's coming along here any day. Hopefully it will be up on bioRxiv. Yeah, good question.
All right, maybe I'll ask my question. So I'm interested in insect-plant interactions and how crop domestication shapes those. And I was just wondering whether in your phenomics pipeline, whether you're looking at any traits that are related to defense, and whether that, if you know of anybody that has that as part of their radar while doing kind of, intense rounds of selection. Because, people have talked about them, and then certainly there has been evidence, that, there's kind of, issues around kind of linkage between traits for, that are kind of inversely linked. So traits for yield may be inversely linked with traits, that are associated with defense.
Yeah, it's a great question. And the short answer is we are not directly measuring defensive compounds using the techniques that I mentioned. However, this is a big issue, particularly for Silphium integrifolium, which has, I don't know if you know David Van Tassel or who have ever heard him. He works for the Land Institute, talk about the struggles of Silphium. So Silphium is a native North American prairie species that is very drought tolerant, and it produces, it's a sunflower relative, and it produces seeds that people are using for oil. And they planted it in experiments in Kansas, and the onslaught of pests and pathogens that followed was just really intense, and it varied from one year to the next what the problem was. So the rust and the caterpillars and the fly, I mean, it's one thing after the next. And they, so particularly in that species, breeding for pest and pathogen resistance and really structuring ecologically, but agriculturally, a system, I think that's the challenge, because in nature, the demographics of these species are such, I think, that they've evolved to help resist infestation. But when you put a bunch of plants in pretty tight proximity, even if they are in a polyculture, it's just a difficult situation. So for that one species, we are in our work measuring disease incidences. And I think I showed some relationships between some of our early stage measurements with particularly rust prevalence in the field. So I guess what we're showing is some relationship between traits that maybe could be useful, but as far as a long-term solution on how to address that, I think that's still a little bit up in the air. I could also, in addition to David Van Tassel, I could also point you to the work of Damien Rivetta and Alejandra Vilela. They have done some really beautiful experiments with Silphium, looking at various morphological and physiological traits, as well as disease incidents.
My former grad student is actually now at the Land Institute. His name's Chase Stratton. So he is active in some of the Silphium work. But yeah, I think in general, there's such a large gulf between kind of, plant, crop genetics and entomology that is unfortunate, but it would be good to have more integration early on as well.
I totally agree. And it's for something like Silphium, I mean, for everything, but particularly for Silphium, it has to happen. I mean, I don't know if Chase is doing that out there now, but it just has to happen.
Mm-hmm. Yeah. All right. Thank you.
Yeah. And if you have ideas of how to do it I'd love to hear more about it, because we recognize that we're just not there yet.
Yeah. For my PhD, I actually worked on sunflowers and kind of how crop domestication shapes species interactions, in this case, tritrophic interactions. So, that's a relative and stuff. So I have a guess as to some of the things that might happen. So welcome to chat anytime.
Yeah, I love that.
Okay, sounds good. Yeah, take care. Thanks.
So I want to respect your time, of course, but would like to ask if you have time for one more question, specifically Ryan's question that's in the chat.
Oh, sure. Here, let me pull it up. Okay, let's see. So I don't know much about this field, but I argue prehistory humans had time to look at perennials for their yields and to choose to domesticate annuals. Today is the goal to GMO perennials to get energy allocation to focus more on reproduction. That's a really good question. So as far as like the question of whether humans actively domesticated or actively did not domesticate, actively domesticated annuals over perennials or excluded perennials, there was a wonderful paper, it's one of my favorite all-time papers, written in the journal Evolutionary Applications in 2010 by David Van Tassel, who I just have mentioned already. That guy's work is amazing. That basically asks why the perennial herbaceous species were not domesticated. And he pitches lots of hypotheses and sub-hypotheses for why that is. My personal take on it is I think annuals just make more faster, and that was what was appealing at the time. I don't know. As far as how to get to a domesticated perennial, there's a lot of different pathways that are happening. And so there are some, I mean, and it's really encouraging to see so many different people working in this space, applying so many different tools. So there are some people who are attempting to genetically modify or edit or whatever you want to call it, perennial plants to increase yield. There are some folks who are working on doing the reverse with annual plants to essentially perennialize them. There is what's called wide hybridization happening, where crosses are being made into an annual background or into a perennial background to get the traits of the other that you want, but still retain the features of whatever it is that you started with. And then there's this concept of de novo domestication, where people are actually going into wild species. And this is kind of where we've been most interested in, going into wild species, perennial species, and looking at what might be a good perennial crop candidate species, and beginning kind of the pre-breeding, it's kind of pre-breeding processes that might get that going. So there's a lot of different paths, that's what I'm saying. And there's no one right one. I think it's just a really exciting area of work for those of us who are interested in the potential perennialization of agriculture. Okay, thanks, Eric.
I'm going to get off now. Thank you for your talk.
Thank you. Thanks for coming.
Thank you so much, Allison. It was so insightful, and it was such a delight to have you
Oh my
talk to all
gosh.
of
Well,
us.
thank you. Well, I would love any feedback anytime, and happy to take any questions whenever.
Super. Thank you so much.
Thank you.
Have a good weekend.
Yep, you too. All right, see you guys. Thanks.
I'll work on the strategic plan soon.
Okay, it's due in 2 weeks.
I know. There's so many other things that are due.
I know. Well, we appreciate your contributions, Eric, in whatever form they come.
Trying.
Okay. All right. I'll see you later.
Thank you. Bye.
Bye.